Welcome back to this next video in which we are discussing multiple sclerosis. So we're currently in the process of discussing the pathogenesis of multiple sclerosis, and the point in the story that we've got up to so far is we have seen how you get activation of the autoreactive clone of CD4 positive T cells. We now want to turn our attention to how the clone of autoreactive B cells is going to be activated. So, cross-reactivity is going to play an extremely important role here again. So here is our pathogen that's infecting some tissue within the body, and here's the incredibly important antigen, and remember this antigen has a peptide fragment that is equivalent to the peptide fragment in the CNS myelin protein that we developed an autoreactive clone of CD4 positive T cells against, and it also has an epitope that is equivalent to an epitope within the CNS myelin protein that we have developed a clone of autoreactive B cells directed against. So, we now want to understand how the presence of that epitope in this antigen is actually going to lead to the activation of that autoreactive clone of B cells. So we want to see how a B cell is activated. So again, allow me to say that this pathogen has a huge number of different antigens. Those antigens all have multiple different epitopes, and therefore, in reality, we will be activating a huge number of different B cell clones in response to this pathogen. So in fact, you're going to get multiple different B cell responses all occurring concurrently in response to this pathogen. Again, we are only going to study one of those B cell responses. We're only going to study the autoreactive one because that's the one we care about for understanding the pathogenesis of multiple sclerosis. But be aware in the back of your head that lots of concurrent B cell responses directed against different epitopes will also be being initiated. So, how do you activate B cell responses then? Of course, the B cells are in lymph nodes. The autoreactive B cells of this autoreactive B cell clone, they're present in lymph nodes. How do you actually activate them? Well, stage one in activation is that the epitope that they are directed against has to actually arrive in the lymph nodes. How does this occur? Well, it occurs because the phagocytes at the site where the infection is actually occurring, when they are breaking down pathogens, so neutrophils, macrophages, and dendritic cells, when they break down pathogens, some of the proteins they release into the extracellular fluid so that they can drain in the lymphatic vessels to the lymph nodes so that we can get the activation of B cell responses. So I'll say that again. Macrophages and dendritic cells, we know that they do this antigen presentation. They break down antigens into little peptide fragments and put them on the surface attached to major histocompatibility complexes to activate T cells. But all of the phagocytes, not just macrophages and dendritic cells, but also neutrophils, when they are phagocytosing pathogens, sometimes they let the proteins go. When they've broken down the pathogen, some of the proteins they chuck back into the extracellular fluid so that those proteins can drain in the lymphatic vessels to go to the lymph nodes so that B cell activation can begin. So let me show this on this diagram here. So we'll put this here, and again I'll remind you we're only going to follow it for this epitope of the antigen. So, the phagocytes, the macrophages, the neutrophils, and the dendritic cells, they're going to release the antigen of the pathogen here into the extracellular fluid. So here is this antigen, and we're really interested in just one epitope of this antigen. So let's say it's this corner down here. This is the epitope that we have a clone of autoreactive B cells that is targeted against that, and that's the epitope that's also present within the CNS myelin protein. Okay, so what's now going to happen then? This antigen, or these antigen molecules, because in reality you've got loads of pathogens, loads of phagocytes, so lots of release of these antigen molecules. These antigen molecules are now going to drain in the lymphatic vessels to the draining lymph nodes. So this is now going to go to the lymph nodes. So I'll put this here. The antigen will end up going to the lymph nodes. And of course, in the lymph nodes, there may well be present examples, members of this autoreactive clone of B cells. So let's bring the star of the show now in. So here is one of these autoreactive B cells. So again, I'll remind you, we're only following one B cell clone response, the B cell clone that's directed against this epitope of this antigen that will be able to cross-react with an epitope of the central nervous system myelin protein. So, how then is the epitope arriving near this B cell? And I'll just label up this B cell. So this is the autoimmune. This is a member of the autoimmune clone. Autoimmune, and at the moment it's naive. So it's an autoimmune naive B cell here. So we have its epitope arriving in the lymph node that it's sitting in here. How is this actually going to end up beginning the activation process of this B cell? Well, the B cell recognizes its epitope, but how does it recognize its epitope? Well, the reason is that on its surface it has examples of its antibody molecules. So remember, we've talked about how all the different B cell clones are directed against different epitopes. The reason they're all directed against different epitopes is that they have a certain design of antibody associated with them, and this antibody can bind to the epitope that they are directed against. So when we talk about B cells being directed against an epitope and we talk about them having an antibody that's built against that epitope, those are one and the same thing. The fact that they have an antibody design that recognizes that epitope is the reason that they're directed against that epitope. So let me show you the antibody molecule of this B cell here. So I'll draw it bound to the surface here. So if you aren't familiar with antibody molecules, remember they have a Y shape, like so, and they're made out of 4 separate proteins. So I'll just go over the names of the separate proteins. So you can see that I've drawn 4 separate lines here. 1, 2, 3, 4. 2 of the lines are big. This one and this one are the big ones. These are what we call the antibody heavy chains. And I've just realised I haven't actually put the word antibody. I don't think I've ever wrote that term before, so I should just introduce you to that term if you're not familiar with it. So antibody is what B cells produce. It's a major molecule of the extracellular adaptive immune response, and for short we abbreviate it down to AB, like so. So these 2 proteins are both antibody heavy chains. Every B cell clone has a certain design of antibody heavy chain, and the 2 antibody heavy chains that it's using here to make an antibody molecule, they're absolutely identical. So it makes 2 identical antibody heavy chains of its own personal design and uses them to make an antibody molecule. The other 2 proteins, this one here and this one here, they're also identical, and they're what we call the antibody light chains. And again, each of the B cell clones will have its own design of antibody light chain. So all of the different B cell clones in your body, they design a light chain, they design a heavy chain, and therefore they have a design of antibody molecule. And the bit that varies is the bit right at the tip here. Most of the heavy chain and light chain is identical between different B cell clones, but the bit that varies is the bit right at the tip, and this is the bit that can bind to the epitope of a protein. So we call this the antibody binding site. So this little bit here, this is, sorry, not the antibody binding site, the antigen binding site. So this is the bit that can bind to an epitope of an antigen. So right at the tip, that's the bit that varies between different B cell clones. Now, B cells can make a form of the antibody heavy chain that is longer than normal and which allows them to implant the antibody molecule on their cell surface membrane, like so. And when they do this, it's known as a B cell receptor, or a BCR for short. So that's what I'm drawing here. This is an antibody molecule that's been given special legs here to allow it to sit in the B cell membrane and act as a B cell receptor. So all the naive B cell clones, they all have their special design of antibody on their surface as B cell receptors, and they don't just have one of these, they have multiple of them. They're covered in them. So that means that they have their antibody, which of course has 2 antigen binding sites for the special epitope that they are directed against, on their surface, and this is how they can recognize the epitope when it arrives. So when this antigen here with the special epitope arrives in the lymph node where this autoimmune naive B cell is, it's going to be able to bind to this antigen's epitope using its B cell receptor. So this is going to come and bind here. So, the question now is what happens next. So, if you were naive here, you might think, well, that's going to be it. Now the B cell is going to say, oh my goodness, my antigen has arrived, here is my epitope that I'm directed against, I now need to activate. I should divide and divide and divide to clonally expand, and then I should start to differentiate into effector B cells, and we should do whatever it is that we do. We need to activate. However, remember what I've already told you. B cells cannot activate that easily. B cells are not allowed to activate that easily, and the reason is that B cell responses are incredibly dangerous. So you have to have a double check here. You have to have a checkpoint to make sure that only B cells that are absolutely necessary to activate get activated, and that checkpoint requires help from CD4 positive T cells, effector helper T cells. So B cells cannot activate yet. This B cell is not going to start dividing, not until it's had the go signal from an effector CD4 positive T cell. How does it get help from an effector CD4 positive T cell? Well, in order to get help, it has to prove to an effector CD4 positive T cell that they are on the same side, they are fighting against the same antigen. It has to prove that the epitope that it's directed against is part of an antigen that the CD4 positive T cell is directed against a peptide fragment of. So it has to chop up the antigen into peptide fragments and present it to CD4 positive T cells, effector CD4 positive T cells, and if that peptide fragment, well, if one of the peptide fragments that it presents matches the peptide fragment that a CD4 positive T cell is directed against, the CD4 positive T cell says, go ahead, you are safe, we are on the same side, we are fighting the same antigen, you can initiate, you can activate, you can do whatever you do. Okay, so this is what's going to happen then. Let's draw the next stage in the story. So coming down here. So here is our B cell. Again, here is its massive great nucleus. And what's going to happen is it's going to endocytose the B cell receptor bound to the antigen that it's directed against an epitope of, and it's going to chop up that antigen. So what's going to happen then? Here is the endocytic vesicle, the vesicle after the endocytosis has occurred. And what I will draw inside this vesicle is just the antigen that this B cell is directed against. So in purple there, that is representing the antigen. Okay, and what it will now do is break down that antigen into loads of little peptide fragments, and it will put those peptide fragments on its surface bound to MHC class 2. Remember, it needs to interact with CD4 positive T cells, effector CD4 positive T cells. Effector CD4 positive T cells work with MHC class 2, not MHC class 1. So B cells only ever use MHC class 2. So I'll put this here. So in red here, this is representing a massively enlarged MHC class 2 molecule on the surface of the B cell. And what it's going to do is it's going to chop up this antigen and put peptide fragments on MHC class 2 molecules. And of course there are loads of different peptide fragments it can create, so it'll end up with loads of MHC class 2 peptide fragment complexes on its surface with different peptide fragments. Also be aware that there isn't just one antigen molecule coming to the lymph node. There's lots of these antigen molecules coming to the lymph node. So it's likely that lots of the B cell receptors will actually capture the antigen, and you'll endocytose it all, and you'll break all of them down, and therefore you'll actually end up chopping up the antigen in loads of different ways. So you'll end up with lots of the varieties of peptide fragments that you could have got from this antigen on your surface bound to MHC class 2. So, what has to happen now? As I've previously said, it needs to now find a CD4 positive T cell that is directed against one of these peptide fragments that it now has on its surface bound to MHC class 2. Now, you might be tempted to say here, well, look what we have over here. Here are the autoreactive effector CD4 positive T cells that are directed against a very special peptide fragment of the antigen, specifically the peptide fragment that was also present in CNS myelin protein. They fit the role perfectly. They're directed against the peptide fragment of this antigen, and indeed they would be able to now go and activate that autoreactive B-cell clone if that autoreactive B-cell clone had been lucky enough to chop up one of the antigen molecules in the correct way to get that peptide fragment and put it on its surface bound to MHC class II. However, the likelihood is that this is not the only clone of CD4 positive T cells that can help in the activation of the autoreactive B cell clone. The likelihood is that when you are fighting this pathogen, when you're initiating CD4 positive T cell responses against this pathogen, this antigen can be chopped up in a huge number of different ways to yield a huge number of different peptide fragments. So the likelihood is that you have initiated many different CD4 positive T cell clone responses concurrently against the different peptide fragments of this antigen. Most of them I did not care about because they were not directed against a peptide fragment that was also present within a self-protein, i.e. they weren't autoreactive responses at all. We only talked about the autoreactive one down here. However, those other ones directed against peptide fragments of this antigen that are not present in self-peptides, they will also be able to activate our autoreactive B cells. So don't forget that those will be important potentially as well. It might not just be the autoreactive CD4 positive T cells that can activate the autoreactive B cell clones. So you might then fairly ask, well, why does it actually matter about having these autoreactive CD4 positive T cells? If their job is just to help with the activation of the autoreactive B cells, you've just told me that we don't actually need the autoreactive CD4 positive T cells. We can use these non-autoreactive CD4 positive T cells that happen to be fighting a peptide fragment of this antigen. Why did you need these to develop multiple sclerosis? Surely all you need is an autoreactive B cell response. Well, you will see how these play a role much later, how they're very, very important in the maintenance of the autoreactive adaptive immune response later on, and how they're also important in causing the white matter lesion inflammation. Effector helper T cells do more than just helping B cells activate. They play other roles as well. So these are going to be important in white matter lesion formation. So wait up for that. So I've just explained something quite complicated to you, so I'll just explain it again to make sure you understand what I'm saying. In order for this B cell to now activate, it needs to receive help, signaling, activation from an effector CD4 positive T cell that is directed against a peptide fragment of one of these antigens that it has on its surface bound to MHC class II. You have potentially initiated a lot of CD4 positive T cell responses concurrently against different peptide fragments of this antigen. Most of those will not be autoreactive. They'll be directed against peptide fragments of this antigen that aren't in any of your own proteins. Yes, we talked at length about that special CD4 positive T cell response that was directed against that special peptide fragment that was autoreactive, but be aware that there are all the other ones as well, and that they are just as good as at activating the autoreactive B cell clone as the autoreactive CD4 positive T cell response would be. Then you may well ask, well, why did you need the autoreactive CD4 positive T cell response to get multiple sclerosis? You'll see that later on. They play a role as well as the B cells in actually forming the white matter lesions. They also play a very important role in maintaining the autoreactive adaptive immune response, because this antigen is going to initiate the adaptive immune response. As far as maintaining it for the rest of your life and maintaining the disease, therefore, is concerned, you do need the autoreactive CD4 positive T cells, because later on in the maintenance of the autoreactive adaptive immune response, it's going to be the actual myelin CNS protein that is causing the maintenance, and the other non-autoreactive CD4 positive T cells will not be effective at maintaining it just with the myelin CNS protein, but we'll go into that much more later on. Okay, so let's now see then what happens once we've got B cell activation. So one of the effector CD4 positive T cells is going to find this B cell, it's going to come across it, it's going to say, look, on your surface you have an MHC class 2 peptide fragment complex with the peptide fragment that I am directed against. You are a good B cell, we are on the same side, we are fighting the same antigen, you can activate. And the chances are that this B cell will come across one of the effector CD4 positive T cells directed against one of the peptide fragments of this antigen because of the fact that we produce so many effector CD4 positive T cells. We've just discussed how there are loads potentially of these CD4 positive T cell responses directed against peptide fragments of this antigen. They will all have produced a huge number of effector CD4 positive T cells. Those will not just have stayed in the lymph node where they started off, those will have gone into the bloodstream and distributed to other lymph nodes, so they'll now be everywhere, all over the body. So it's very likely that in the lymph node where this B cell is, it will be able to find an effector CD4 positive T cell directed against one of the peptide fragments that it is trying to fight an antigen that contains that peptide fragment. So let's put this in here. So it will bump into one of these helper T cells, effector helper T cells, directed against one of the peptide fragments of the antigen, and it will give this B cell permission to activate. That's the safety check. Once the helper T cell gives the B cell permission to activate, the B cell is then going to activate. How does it do this? Well, of course, it divides and divides and divides. So let me draw this here. So it's going to expand the clone. And again, I'll just draw 5 of them, but in reality it will divide much more than to produce 5. It will divide and divide and divide and produce a huge, great population of these B cells now. After division has occurred, you should be getting fed up with this story. It's exactly the same as what happened in the T cell activation. The next thing to occur is differentiation. So the progeny of this B cell that's activated are now going to differentiate. And again, don't forget the fact that when you differentiate, there are 2 different types of cell that you can produce. There's not just the effector B cells, there's also the memory B cells. And I'm going to actually start off with the memory B cells, and then we'll go on to the effectors. So again, this is the minority that will differentiate into memory B cells, but we'll start off with them. So let's have 2 here, differentiating into memory B cells. So these 2 are going to be memory B cells. And again, the purpose of these is exactly the same as the purpose of the memory CD4 positive T cells. They are to replace the original naive one. So they will go up here and replace the original naive one. And again, I'll give you that explanation as to why we need these. Think about this again, not in the context of autoimmune disease. Think of it in the context of an immune response that is fighting a pathogen. This is our naive B cell here. It's directed against an epitope of the antigen of this pathogen. It might be the case that we only have 1 B cell in the body that actually is directed against that epitope. Now we're going to use it. Here is the epitope. The antigen has arrived. We want to use it. It's going to divide and divide and divide. If they all differentiated into effector B cells, what would happen? Effector B cells do not live that long, nowhere near as long as naive B cells. So within a few weeks to months, all the effector B cells would have gone, and then you would have lost the entire clone. Of course, the bone marrow is still producing new B cells, so there is the possibility that we would create it again, but it's a chance, basically. We might have lost a very good clone of B cells that we might need again. I mean, we might get infected by this pathogen again, and therefore it would be very handy to have this clone again to use. So we have to replace the naive B cells. We have to create some more B cells that can be activated if we ever need them again, i.e. if we ever face this epitope again. And this is what memory B cells are for. So these memory B cells are going to replace the original naive B cell, and they can do the exact same thing as the naive B cell, i.e. if the antigen comes along in the future, they will bind to that antigen, they'll be partially activated by that antigen, they'll do this process again, they'll endocytose the antigen, break it down into peptide fragments, display it on their surface, bound to MHC class 2, and then they'll need help from effector CD4 positive T cells directed against one of the peptide fragments of the antigen, and then they can divide and divide and divide, and again their progeny can differentiate into either memory B cells to replace them, or effector B cells. So they replace the naive B cells, and they are really important in being able to continually initiate more of the immune response because of the fact that effector immune cells do not live forever. The effector CD4 positive T cells and the effector B cells do not live forever. They live a very short time compared to the naive ones, and the memory ones can last your entire lifetime. So if you want to ever be able to initiate this response again, you need memory cells. And to justify a disease like multiple sclerosis, where this autoimmune attack is going on for the rest of your life, we have to understand the memory cells, because they're the reason that the immune response can continuously be renewed and maintained. So it is very important to understand about the memory cells. So now let's talk about the effector B cells. So another colour. So we'll go to yellow here. So the effector B cells have a different shape. They have a more oval shape, like so. And the majority of our B cells are now going to differentiate into effector B cells. So these cells here, these are effector B cells, and you probably are aware that effector B cells have another name, a name that's more commonly seen. And they're called plasma cells. So these are the major cells of the B cell response. So what are the plasma cells now going to do? So the plasma cells will go into the blood, and they will then go to the bone marrow. The bone marrow is the place where plasma cells like to hang out. So these will go to the bone marrow, and they will live their relatively short lifetime of a few weeks to months in the bone marrow. And whilst they are in the bone marrow, the way that they are going to contribute to the immune response is they are going to start up massive production of soluble antibodies. So coming back up to antibodies up here. So we drew an antibody here with the 2 heavy chains and the 2 light chains, the classical Y shape. In this picture it's a little bit upside down, but it is the classical Y shape. We drew an antibody where the heavy chains had got this extension on them that meant that they could be implanted into the cell membrane. Most antibody molecules do not have those very long heavy chains. They're made with shorter heavy chains, so that instead of sitting in the plasma membrane, they're secreted into the extracellular fluid, and we call these soluble antibodies. So our plasma cells are going to go into the bone marrow, and they're going to start chucking out antibody molecules, soluble antibody molecules. These antibody molecules will go into the bloodstream, and they will then leave the bloodstream at the site of inflammation, i.e. where the infection of with the pathogen is occurring, and there they will be able to bind to the epitope of the antigen that they are directed against. So I might even just draw this in here. So here's our pathogen, and here now is an antibody molecule directed against that epitope there, coming along. They'll be able to bind to that epitope, and the way in which they contribute to the destruction of the pathogen is firstly, well, there are lots of different mechanisms. Firstly, if this protein is extremely important in the life of this pathogen, then binding this great big antibody molecule to it might make this protein dysfunction, and therefore it might interfere with the ability of the pathogen to live. In addition, these antibody molecules activate phagocytes to destroy the pathogen, and this is a process called opsonization. So I'll write this big term here. So to justify this, you have to understand that phagocytosis is not something that's ridiculously easy. Neutrophils, macrophages, and dendritic cells, these are the examples of phagocytes that we have seen. How do they decide what should be phagocytosed and what should not be phagocytosed? Because they can't just phagocytose any old thing, because they'd end up phagocytosing perfectly healthy tissue. They have to be very sure that what they are about to phagocytose is something bad, potentially that it's a pathogen or that it's a dying part of a human cell. So they look for markers, molecular markers, such as pathogen-associated molecular patterns, to decide whether the thing they've come into contact with should be phagocytosed. But it is a and ahhing moment. They are sort of sitting there saying, is this thing abnormal enough to phagocytose? And sometimes they'll get it wrong. They'll say they've got a pathogen sitting right in front of them and they don't have enough proof that it is a pathogen for them to actually phagocytose it, and therefore they decide not to phagocytose it. And that is important, because if they phagocytosed anything that looks mildly abnormal, they would end up phagocytosing a lot of healthy tissue. So it is important that they have to be very sure that what they're about to phagocytose is abnormal before they do it. This is where antibody molecules can be extremely helpful, because if loads of antibody molecules are bound on the surface of a pathogen, that is an absolute giveaway sign that this thing is abnormal. If you're covered in antibody molecules, you are not normal, and therefore it tells phagocytes that this thing is something that they can phagocytose, so it hugely aids phagocytes deciding what is abnormal and what is not, producing antibody molecules. And this process is called opsonization. In particular, on the surface of macrophages and dendritic cells, there is a receptor known as the Fc gamma R3 receptor that can bind to antibody molecules. And I should just say a little thing that I've missed out in this story. So, there are different forms of soluble antibodies that you can produce. So let me go back to my picture of the antibody up here. In fact, I'll draw a new picture of an antibody. I want to draw it the right way up this time so that you can actually see it looking like a Y. So here are the two heavy chains and here are the two light chains. And remember, there are little connections. There's a connection there between the two heavy chains and connections between the heavy and light chains, like so. So here's an antibody molecule. Remember, the bit that's unique to the clone of B cells is this bit right at the tip, the tips of the heavy chains and the tips of the light chain. The rest of the heavy chain and light chain isn't unique. That's the same that all the different clones of B cells will use. However, these lower down bits of the heavy chain, you can use different versions here. There are different versions of the heavy chain that each clone of B cells can decide to use when it's making antibodies. For instance, it can decide to make very long heavy chains where it can then implant the antibody molecule into its cell membrane to create a B cell receptor, as we previously described. But even when it's deciding to make a soluble antibody, it can choose different bits to go in here. There aren't that many choices for it, but there are a few different choices that it can put in here. So it's got its unique design up here, but it can choose to have with its unique epitope recognition domain up here different bits in this bit. And this lower down bit of the antibody is called the Fc region. Fragment crystallizable, I think, is what it stands for. But this lower bit of the antibody, know that this is the Fc region. In fact, I'll give you a little bit more terminology. So the Fc region is, strictly speaking, all of this portion of the antibody. And then right at the tips, of course, those are the bits that actually bind to the epitopes. Those are the antigen binding sites, and we call those FAB, which stands for fragment antigen binding. So, we can split antibodies into the Fc region and then the two FAB regions. Now, what I am telling you is that the Fc region, there are different variants of the Fc region. Not many, but each B-cell clone, when it's deciding to make soluble antibodies, it obviously has its set FAB up here, but it can choose from a few different variants of the Fc region down here. And the one that it chooses in the autoreactive B-cell clone that is going to occur in multiple sclerosis is the most common type in the blood, one of the most common types to be made, which is called IgG. So, what this stands for is the Ig stands for immunoglobulin, which is another name for an antibody. It's the fancy name for an antibody. And then G tells you that this Fc region is specifically the one that we call Fc gamma. And I'm telling you all of this just to justify the name of that receptor. So let me summarize this complicated thing that I've been trying to tell you here. So we have discussed how the adaptive immune system is very complicated. There are loads of different B-cell clones, and all of the different B-cell clones have their own FAB region for antibodies. The Fc region, however, when a certain B cell clone is deciding to make an antibody molecule, there are different options for what Fc region it can put in here. Not many, but there are some. One of the Fc regions that it can put in is called an Fc gamma region, and then the antibody molecules that you produce then are known as IgG molecules. There are other types that you can put in here, such as IgM is another form of immunoglobulin molecule, or IgA is another form as well, and IgE is another famous form. Okay, but IgG is the form that's going to be made in this autoreactive B cell response in multiple sclerosis. It's not particularly well understood why some plasma cells choose to make certain forms of soluble antibody molecules, i.e. I can't really provide you with a justification as to exactly why these plasma cells that are these autoreactive plasma cells are specifically going to choose to make the IgG molecule. We think it must have something to do with the actual pathogen that they were trying to fight up here. So something about this pathogen meant that the B cell response decided that the most appropriate type of antibody molecule to create was IgG. So be aware that the type of antibody molecule that's going to be secreted by these autoreactive plasma cells here that have been initiated in response to this pathogen is the IgG type. Now, why did I tell you all of that? Why did I make it so much more complicated? Because I wanted to justify this receptor's name. Fc gamma refers to the fact that these antibody molecules here are going to be Fc gamma type antibody molecules. R3 just means receptor type 3. So on the surface of the macrophages and indeed the dendritic cells, we have this receptor. Here it is. This is the Fc gamma R3 receptor, and this can recognize the Fc gamma portion, the fragment crystallizable gamma portion, this lower portion of the antibody molecule, and therefore it can bind to the antibody molecule when it's coating a pathogen, and that's the way that it can recognize these pathogens that are coated with the antibody molecules. So that's one of the key ways that antibody molecules can help in the destruction of pathogens. In addition, we talked, when we were talking about the inflammatory response, about how there are lots of proteins in the blood that come out in an inflammatory exudate, which help to attack pathogens. These proteins can also be recruited by antibody molecules to attack the pathogens that they are stuck onto. So antibody molecules are incredibly good at helping to attack pathogens. So, that's sort of like the motivation for why you produce antibody molecules against this pathogen. Let's now see where we have actually got in the story, and then we'll take a break, and in the next video we'll actually start seeing how white matter lesions are going to form. So the point that we have got up to now is that we have successfully activated the autoreactive CD4 positive T cell clone and the autoreactive B cell clone. We now have loads of effector CD4 positive T cells directed against the peptide fragment that is present within our CNS myelin protein, and we also have a whole bunch of plasma cells that are directed against the epitope that is within the CNS myelin protein that are sat within the bone marrow, but which are chucking into the blood antibody molecules that are directed against that epitope of the CNS myelin protein. So we have effector CD4 positive T cells in the bloodstream that are directed against the peptide fragment of the CNS myelin protein, and antibody molecules in the bloodstream directed against an epitope of the CNS myelin protein. In the next video, we will see how these things are going to actually lead to the formation of white matter lesions within the brain and spinal cord.